Hello class, I hope you all are in good health, and today in this video lecture we are going to start oogenesis. Okay, so previously in this chapter we have seen the male reproductive system followed by the spermatogenesis, that is the formation of a sperm, and then we studied the female reproductive system. Okay, and in this video lecture we will study the formation of female gamete. Okay, and this process is called oogenesis. Okay, so if you have to define oogenesis, we define it as the process of formation of matured female gamete. Well, so the process of formation of matured female gamete by a adult human female is called oogenesis. Okay, now if we compare the process of oogenesis with spermatogenesis, okay, we find that there is distinct difference between the two. Okay, why we want to compare? We want to compare because both of these processes are a type of gametogenesis. Okay, spermatogenesis and oogenesis both are type of gametogenesis, that is, these are the processes by which gametes, okay, male and female gametes, are produced by human male and female respectively. Okay, so what is the difference between the two? The major difference between spermatogenesis and oogenesis is that the spermatogenesis is initiated only after puberty. Okay, so before puberty there is no spermatogenesis. The process has not been initiated at all in a male. Okay, now if we look at the oogenesis, okay, the process of oogenesis is initiated in the embryonic condition. Okay, so when a female is still in the womb, she has not taken birth. Okay, in her fetal condition, in her fetal stage, in the ovaries, okay, when the ovaries are developed in the fetus, okay, female fetus, at that point only the process of oogenesis is initiated. Can you see? So this is the major difference in oogenesis and spermatogenesis. Oogenesis starts in the fetal stage, in late embryonic stage. Okay, whereas spermatogenesis only initiates after puberty or at puberty. Okay, apart from that there are other differences as well, such as we will see that in spermatogenesis, in the process of spermatogenesis, both meiosis 1 and 2 occur back to back. Okay, so there is no arrest, no delay. The primary spermatocyte undergoes meiosis 1, leading to formation of secondary spermatocytes, and then secondary spermatocyte undergo meiosis 2 without a delay, right, leading to formation of spermatids. And then we saw that spermatids transform or metamorphoses into spermatozoa or sperm, right? So there the meiosis 1 and meiosis 2 are taking place back to back. There is no delay. However, in case of oogenesis, we will see in this video lecture that meiosis 1 and 2 does not happen back to back. There is a gap between the two. Also, meiosis 1 as well as in case of meiosis 2, and both the cases, the cells are arrested for a very long time in their prophase. Okay, so cells are arrested in prophase 1. After that, the meiosis 1 is completed, and then again the cell is arrested in meiosis 2, especially prophase 2 of meiosis 2. Okay, so here in this case the meiosis is not continuous, rather there are gaps in between. Okay, the cells remain arrested. So we will see there are a lot of differences between spermatogenesis and oogenesis. Okay, so let us start the process of oogenesis. Well, so we will start from the embryonic stage when this process initiates. Great. So what we find is, in the fetal conditions, okay, when the ovary is being developed, there is a lining of epithelium called germinal epithelium. Okay, let me draw it for you. So this is germinal epithelium. Can you see? This is a layer of germinal epithelium, which is mainly composed of simple cuboidal epithelium, and these are present as a lining of the ovary. Okay, now these cells are limited in number, and the cells of this germinal epithelium are known as primordial germ cells. Okay, these are primordial germ cells, and these are diploid. Now, in these diploid cells, mitosis will occur. Okay, remember, mitosis, it is not meiosis, mitosis. Okay, so mitosis will occur. As a result, the number of cell will increase, and the new daughter cells formed are called oogonia. These are called oogonia. Okay, do you remember these things? Now, this happens at approximately the 25th week of pregnancy. Okay, so when the female fetus in her mother's womb is of about 25 weeks of age. Okay, so 25 weeks of age, at that point this mitosis will occur in the primordial germ cells, leading to the formation of oogonia. Okay, leading to formation of these new cells called oogonia. Okay, now how many such oogonia are formed? Approximately around 1 to 2 millions. Okay, 1 to 2 millions of oogonia are formed in each fetal ovary. Okay, you can say that 1 to 2 million oogonia per ovary are produced in the fetal conditions. Okay, now if we look this number in terms of lakh, we can say about 10 to 20 lakh oogonia are produced in each ovary. Okay, now after this no more oogonia are going to be formed. Okay, so this is a kind of multiplicative phase. Okay, this is a type of multiplicative phase of oogenesis. Okay, where primordial cells increase their number by mitosis to produce around 10 to 20 lakhs of oogonia, or 1 to 2 millions of oogonia per ovary. Great. Now what happens after this? Can you see? This is the oogonia. Now the cells of oogonia undergo a process of enlargement, so they slightly enlarge in size. At the same time, they undergo a process of differentiation. Okay, now not complete differentiation, and in this stage they will now enter into meiosis 1. So the oogonia are going to enter into meiosis 1, but they are not going to complete it. They will just enter into meiosis 1, and you know that the first stage of meiosis 1 is prophase 1, right? So in prophase 1 they enter into this meiosis, and the first stage is prophase 1, and they remain arrested in this prophase 1. Okay, they are not able to complete it. They remain in the prophase 1. And I hope you remember that prophase 1 is a very long phase. It is further divided into 5 sub-stages. Okay, leptotene, zygote, diplotene, okay, and so on. So there are multiple phases in prophase 1. So they remain arrested in meiosis 1 till the female attains her puberty. Okay, so now she will take birth and she will grow up to the age of like 12 to 14 years, and at the age of 12 to 14 years she will attain her puberty. So what I'm saying is, these Cells which are now arrested in prophase 1 of meiosis 1 will remain in this stage for next 10 to 12 years or maybe 14 years depending on the age of puberty. Okay, now these arrested cells are called primary oocyte. Okay, these are called primary oocyte, and since meiosis 1 has not been completed, still they are 2n. They are 2n. These are diploid cells. Okay. Now, during the fetal stage only, during the fetal stage, these primary oocytes, every single primary oocyte is surrounded by almost a single layer of follicular cells. Can you see this pink one? Okay, so they are surrounded by a single layer of follicular cells, and now this whole structure, okay, this whole structure having this follicular cells and the primary oocyte is called primordial follicle. So this is called primordial follicle. Yaad rahega. And this primordial follicle, how many will be there? Around 10 to 20 lakhs or 1 to 2 millions per ovary will be present and will be waiting for puberty. Correct? So around 10 to 12 years they are going to wait there in this arrested phase. Great. So they will remain in this. Now during this whole process, okay, the whole process of waiting 10 to 12 years or maybe 14 years, it happens that some of these primordial follicles naturally degrade. Okay, so out of 1 to 2 million there will be many primordial follicles which are going to degrade naturally, and at the end when a female reaches her puberty, only about 60 to 80,000 primordial follicles are left in each ovary. Okay, so from 1 million, that is 10 lakhs, how many are left? Only about 60,000. Can you see? So there is a lot, around 40,000. So 20 to 40,000 of these follicles, okay, primordial follicles, degenerate naturally till the female attains her puberty. Now these data are from NCERT, so they are important. Do remember. Okay, so do remember the fact that in fetal stage there are around 1 to 2 million oogonia per ovary, but by the time the female reaches her puberty, only about 60 to 80,000 follicles are left in each ovary. Okay, so per female there will be twice the number of this. Okay, that is 1.2 to 1.6 lakhs. Okay, great. याद रखिएगा इस बात को. Now what happens once the female reaches her puberty? She will start the menstrual cycle. Okay, and the first menstrual cycle of a female is called menarche. Okay, this is called menarche. Okay, this we will see later once we will study the menstrual cycle, but I hope you all know from class 10th that the first menstrual cycle of a female's life is called menarche, and the time when it ceases is called menopause. Correct? Now, so what happens now is from puberty up to the age of menopause, one primordial follicle in each ovary starts to develop into a matured follicle towards the formation of female gamete in each ovary alternately. When I say alternately, it means, so suppose in 1 month the right ovary started the process, so in the next month left ovary will do. Okay, then next to next month again the right left, this goes on. It seldom it will happen that both ovaries will start to develop its follicle in the same time, okay, or in the same month. This usually does not happen. Usually what happens, one primordial follicle will start to grow each month, okay, in one of the ovaries in a female, okay, and during this whole process this primordial follicle will develop into a matured follicle, and from matured follicle ovum will be released by the process of ovulation, and finally the female gamete, that is egg cell, will be formed. This we will see later, okay, in this lecture. In later section we are going to study how this will happen. Great. So alternately, both ovaries do not start the process simultaneously. Do remember this. Great. Now what happens, let's see. So in puberty, after puberty, in 1 month the ovary have this primordial follicle, right? So this is primordial follicle. Now what happens is this primordial follicle starts to develop, not all at once, one at a month, okay. So some primordial follicle may have to wait up to like 40 years or 45 years of age. Some may only have to wait 14 years, okay. It depends which starts the first. So primordial follicle, one primordial follicle out of this 60 to 80,000 starts to grow, and once it starts to grow, the first thing that will happen, the follicular cells arrange outside the primary oocyte, okay. So these follicular cells arrange, the follicular layer will going to be more than two or more than two layers, okay. At the same time the primary oocyte is going to secrete a non-cellular fibrous layer, okay, of glycoprotein. So this non-cellular fibrous layer which is secreted by this primary oocyte is mainly made up of glycoproteins, is secreted just outside this primary oocyte and forming a protective layer called zona pellucida. Okay, so can you see, this is primary oocyte, and outside the primary oocyte this non-cellular layer called zona pellucida is secreted, and it is secreted by the primary oocyte, and outside this follicular cells are arranged. Now this stage is called early follicle, okay. Now there is another stage called late primary follicle. In case of late primary follicle, what will happen, the follicular cells will further increase, so there will be multiple layer of follicular cells. That's it, okay. Now what happens further, this primary follicle will grow, okay, grow in size as well as in the structure. What will happen, let us see here. Now this will grow in size, the follicular layer will increase, so the number of layers of these follicular cells will increase, and they will form a membrane, thick membrane called membrana granulosa. Okay, so membrana granulosa in an ovarian follicle is a thick layer of follicular cells. याद रखिएगा इस बात को. Okay, now on the outer side or the periphery of this follicle, there are some cells arranged. Okay, these cells are stromal cells and they form the theca. They form the theca. What they form here is theca interna. Okay, theca interna or internal theca. Great. So this internal theca is formed, but this theca is not well differentiated as of now. Okay, great. The primary oocyte and the zona pellucida will persist. Now this structure, this ovarian follicle with theca interna, after the secondary follicle, this will further grow and become the tertiary follicle. Now, what are the changes that happens? In case of tertiary follicle, if you see here, the theca, the outer theca gets differentiated into two layers. Okay, theca externa, that is external theca, and internal theca called theca interna. Okay, so now theca is differentiated into two distinct layers, theca externa and theca interna, and both of these theca are formed of stromal cells. Okay, these stromal cells are spindle shaped, and the theca interna, the cells of theca interna, or I can say the stromal cells of theca interna, secrete estrogen. They secrete estrogen, which is the female sex hormone. Okay, so some amount of estrogen is secreted by the theca interna, stromal cells. Okay, and now this theca interna is part of this growing ovarian follicle. Hence, we say that estrogen is secreted by the ovarian follicles, in particular the cells of theca interna. Which type of cells? Stromal cells. Do remember these points, they are important. Okay. Now, both these theca externa and interna cells are residing on a basal membrane. Okay. So there is a basal membrane in between the membrana granulosa and the theca. Great. Now, on the inner side of the theca interna, what we find is the follicular cells, right? And these follicular cells are going to form the membrana granulosa, as we have seen earlier. So those structures are going to persist. However, here in the membrana granulosa, there is a pouch formed, and this pouch is called antrum. This is very important. So antrum is formed. What is antrum? It is a fluid filled space. Okay, and the fluid filled in this antrum is called Leaker folliculi or Leaker follicle. Both are correct. Okay, so mainly it is called Leaker folliculi. So there is an antrum that develops in the membrana granulosa. Okay, and this antrum is filled with Leaker folliculi. The formation of antrum is the characteristic feature of tertiary follicle. Remember this. Okay, so the presence of antrum is the characteristic feature of tertiary follicle. This is the structure that differentiate secondary follicle from tertiary follicle. Okay, great. Now, inner to the antrum, again we find a layer of follicular cells around the oocyte. Okay, and on the other side again we find the zona pellucida which persist. Okay, inside zona pellucida there has been significant change in the primary oocyte. Now, during this whole process, when the follicle was transforming from primary to secondary and then secondary to tertiary, the primary oocyte was busy in completing its meiosis 1. Okay, remember meiosis 1 has started before the birth of the child. Okay, in the fetal stage. After puberty, once this particular primary follicle has started to grow and develop, now this will continue the process of meiosis 1. Okay, and it will finish it by the time it will become tertiary follicle. Okay, so when the cell was in primary follicle, it was still arrested in prophase 1. As the follicle is growing, in the meantime the primary follicle is completing its prophase. Okay, metaphase, anaphase, telophase, and then cytokinesis. Great, and thus the meiosis 1 will be completed. Now, this meiosis 1, which has been completed by the time the follicle becomes tertiary follicle, is not equal but unequal. So this meiosis 1 that takes place here in oogenesis is unequal division. Okay, because of this unequal division, the daughter cells, the two daughter cells formed are of not equal size. They are of unequal size. One will be larger and the other will be smaller. Correct? So the two daughter cells formed here, both will be haploid and they will not be equal. The larger haploid daughter cell is called secondary oocyte. Okay, and the second smaller daughter cell formed, which is also haploid, is called the first polar body. याद रहेगा? Great. इस चीज को याद रखिएगा. By the time the follicle becomes tertiary follicle, the primary oocyte complete its meiosis 1. As a result, two cells are formed, secondary oocyte and first polar body, and both of these cells are haploid. Okay, great. Now what will happen? The tertiary follicle will further develop to become Graafian follicle, okay, or matured follicle. Okay, so Graafian follicle or matured follicle are the same thing. Okay, Graafian we call in the honor of the scientist. Okay, so Graafian follicle or matured follicle are the same thing. Whenever in question you find matured ovarian follicle, it means they are referring to Graafian follicle. Okay, and the Graafian follicle is fully developed follicle, ovarian follicle. Well, what changes occur here? Let us see the complete structure of Graafian follicle. What we find is, again, the outer protective layer is theca externa and theca interna. They are well established, well bifurcated. Okay, so you can see them, and they are again formed of stromal cells. They are formed of stromal cells, and these cells are going to secrete estrogen, correct? Especially the theca interna cells. Now on the inner side of this theca we find membrana granulosa, and in membrana granulosa the antrum persists. The antrum is going to persist, rather it is going to further increase. The antrum further develops. Okay, and still this antrum is filled with Leaker folliculi. Great. Now because of this developed antrum, the whole membrana granulosa is differentiated into two parts, right? Outer side is membrana granulosa, and inner side, which is around this secondary oocyte. Okay, now that layer is called cumulus oophorus. Okay, called cumulus oophorus. It is important, beta yaad rakhiyega aap log. It is called cumulus oophorus, and these pink cells, pink small cells that I have drawn are the cells of cumulus oophorus. Okay, now this cumulus oophorus is still connected with the membrana granulosa. Can you see here? With this part, which is known as discus proligerus or simply discus. Okay, so the inner part, which contains the secondary oocyte, is not actually floating in the antrum. Okay, this whole structure is attached with the membrana granulosa by cells of discus proligerus. Okay, so it is a disc-like structure of cells, hence we call it discus proligerus. Okay, great. Now once you see inner side to the cumulus oophorus, what you find is the corona radiata. Okay, corona radiata. Now corona radiata is made up of long columnar cells, okay, which appear to radiate outside, okay, and they look like a crown, hence named corona radiata. Corona means crown, okay, and radiata because they are spike like, they appears to be radiating outside of the oocyte, hence corona radiata, and they are present in 2 to 3 layer of cells. Okay, great. So these are corona radiata. Now inner to the corona radiata we find zona pellucida, which was non-cellular fibrous layer made up of glycoprotein still there. Now the space between zona pellucida and the secondary oocyte, this space is called perivitelline space. Okay, this is called perivitelline space. Can you see this? Well, so there is perivitelline space. Now inner to the perivitelline space you will find secondary oocyte, which is haploid, and this secondary oocyte, after completing its first meiosis, when it becomes secondary oocyte, it will immediately start the meiosis 2. Okay, but again it is unable to finish the meiosis 2. Okay, it will enter into meiosis 2, and the first stage of meiosis 2 is prophase 2, and it will remain arrested in prophase 2. It is unable to complete. Why it is so? Because the secondary oocyte does not have centriole. Okay, so the secondary oocyte does not have its own centriole, and without centriole there will be no formation of spindle fibers. So the cell cannot go into the metaphase. It will remain arrested in the prophase 2. Okay, so this secondary oocyte has initiated meiosis 2 but remain arrested in prophase 2 of meiosis 2. Great, याद रखिएगा इस बात को. ठीक है. Now the first polar body will remain there or it may degenerate. Okay, great. So can you see the whole structure now? So there is theca. Okay, and after theca there is membrana granulosa, then there is antrum, then there is cumulus oophorus, corona radiata, zona pellucida, perivitelline space, secondary oocyte which is arrested in prophase 2. Okay, and then first polar body may or may not be there. It may degenerate or may be there. Okay, and then there's this discus proligerus. Now this whole structure is called Graafian follicle or matured follicle. Okay, now this part, this whole structure is called ovum. Okay, not when it is inside the Graafian follicle. Once there will be ovulation, okay, this outer theca and membrana granulosa will rupture. Okay, and this discus proligerus will detach. As a result, this internal structure having secondary oocyte, first polar body, zona pellucida, corona radiata, and some cells of cumulus oophorus will be released from this Graafian follicle, and this released part which contains the secondary oocyte is called ovum. Okay, let us say, and the process of this release is called ovulation. Okay, where this how this will happen, let us see. So this diagram is from NCERT. If you look at, this is the ovary. Okay, and these are the blood vessels of the ovary, ovarian blood vessels. If you see, now these are primary follicles. Can you see here? This is primary follicle. Primary follicle went into the secondary follicle. This is secondary follicle. Okay, now there is a formation of antrum. Okay, so this is tertiary follicle, and after tertiary follicle it further moved to form Graafian follicle. Now in Graafian follicle, if you see, these are the cells outside, okay, forming the theca. Then there is membrana granulosa cells, there is antrum with liquor folliculi, there is discus. Okay, this is secondary oocyte, and outside secondary oocyte there will be zona pellucida, corona radiata, and cumulus oophorus cells. Okay, so this is the Graafian follicle, and on day 14th of this menstrual cycle, okay, because this whole process of maturation of one primary follicle into Graafian follicle takes about 14 days. This is completed in 14 days. At day 14, this wall of ovary ruptures from here. Okay, at the same time this whole structure of the Graafian follicle ruptures, this discus proligerus will be ruptured, and this structure having secondary oocyte, first polar body, corona radiata, and some cells of cumulus oophorus is released. Okay, now this structure is called ovum. The process of releasing is called ovulation, and this is happening at day 14 of the menstrual cycle. This we will again see later. Now, what is left in the ovary is the membrana granulosa, okay, and the theca cells of the ovarian follicle. Okay, ovum has been released, and whatsoever of the Graafian follicle is left, now this left part will rearrange itself to become corpus luteum. So this will rearrange to form corpus luteum. Okay, let us see. So what happens is, from the ovarian follicle, ovulation will occur at day 14. Okay, it is an average number. So at day 14 ovulation will occur, and as a ovulation, ovum will be released. Okay, but the remaining cells of this Graafian follicle will become corpus luteum. Now this corpus luteum, the cells of this corpus luteum are going to secrete progesterone, a type of female sex hormone which is responsible for maintenance of pregnancy. Okay, now there are two cases. If the female become pregnant, then this corpus luteum will be maintained for next 2 to 3 months. Okay, if not, then the corpus luteum will be maintained only for next 12 to 13 days. Okay, after that, this corpus luteum will start to degenerate. Okay, as a result, the progesterone levels will go down, and the corpus luteum will finally degrade to form corpus albicans. Okay, so corpus albicans is nothing but the scar tissue of corpus luteum. Great. So it depends on the pregnancy. If pregnancy happens, then corpus luteum will be maintained for months. If not, corpus luteum will be degenerated. Okay, degenerated by 27th or 28th day of the menstrual cycle. Okay, now what will happen with the ovum? The ovum, which has been released, contains secondary oocyte, and remember, the secondary oocyte is still in prophase 2 stage. Okay, it has not completed its meiosis 2, right? Why it has not completed? Because it does not have centriole, and without centriole it cannot do the segregation of chromosomes. Metaphase will not be completed, right? Now this ovum will be collected by the fallopian tube, and inside fallopian tube, if fertilization occurs, okay, now we are considering that fertilization has happened. Okay, once the fertilization will happen, the sperm will bring one centriole, one centrosome into the secondary oocyte. Okay, now with the help of this one centrosome that contains centriole, the secondary oocyte will be able to complete its meiosis 2. Okay, and once it will complete this meiosis 2, which is again unequal division, okay, it will form two daughter cells, and both the daughter cells will not be of equal size, right? There will be a larger egg cell and a smaller second polar body. Okay, now this egg cell form will be again what? Haploid. Now this egg cell, the egg cell cytoplasm, also have the nucleus of the sperm. Now both the nucleus of egg cell and the sperm will fuse, and this process is called syngamy. As a result, zygote will be formed. Okay, and after that the zygote will further develop into embryo, then fetus, and finally childbirth will take place. Okay, so this is how it happens. Now the whole perspective that we need to cover here in this video lecture under oogenesis was up to the ovum formation. Okay, how this happens, or maybe up to the egg cell formation. Okay, this is the final female gamete. Well, so is it clear? Let us revise once again what we have seen, that in case of oogenesis, the process starts very early in the life of a female child. Okay, when she is still in her mother's womb, in her fetal condition. Okay, the process of oogenesis is initiated, and at that time the primordial germ cells undergo mitosis to form oogonia. Okay, oogonia is plural, oogonium is singular, and about 1 to 2 million oogonia are formed in each ovary at about 25th week of pregnancy. At the 25th week of the fetus. Okay, great, or pregnancy, we can say. Now, the cells of oogonia will undergo the further development process. They will start the process of meiosis. They will enter into the prophase 1 of meiosis 1, but they are unable to complete it at that time. So they will remain arrested in prophase 1, and they will remain arrested for very long time, till the age of puberty. Okay. Now, during this phase of arrest, many of the primordial follicle or primary oocytes degenerate naturally. Okay, and by the time a female reaches her puberty, only about 60 to 80,000 primary follicles or primordial follicles are present in each ovary. Okay, not more than this. Great. Now what happens? From the puberty up to the menopause, or you can say from the menarche to menopause, okay, alternately one primary follicle or primordial follicle will start to develop each month. Okay, and it will start to develop into matured Graafian follicle, okay, which have the ovum, and finally from ovum the matured female gamete, that is egg cell, will be formed. Okay, so once this primordial follicle starts to develop, it will firstly increase the number of follicular cells. So the outer covering of follicular cells will increase. Okay, at the same time the primary oocyte cell is going to secrete a fibrous non-cellular protective layer around itself, which is made up of glycoprotein, and this layer is called zona pellucida. Okay, yaad rahega. Great. Now what happens? The primary follicle will further grow. Okay, especially it will grow in size because of division in follicular cells. As a result, multi-layered follicular cells are formed. Now this multi-layered layer of follicular cells is called membrana granulosa. Okay, the zona pellucida and primary oocyte will persist in the middle. Okay, at the follicular cells, on the outer side of the follicular cells, a layer of stromal cells are arranged, which form the outer theca. Okay, now this theca is as of now not well differentiated and only forms the theca interna, but there are these spindle shaped cells of stromal cells. Okay, now this stage is called secondary follicle. Further, the secondary follicle develops into tertiary follicle. In case of tertiary follicle, we find that the theca is differentiated into two parts, that is external theca and internal theca, right? And the cells of internal theca secrete estrogen. We find that the follicular, inside the follicular cells, an antrum develops, and this antrum is filled with a fluid called liquor folliculi. Okay, rest of this structure persists. At the same time, by the time tertiary follicle is formed, the primary oocyte has finished or completed its meiosis 1. Okay, as a result, two haploid daughter cells are formed, and these two cells are unequal. Okay, the larger cell is secondary oocyte and the smaller cell formed is first polar body. याद रखिएगा इस बात को. Okay, next what happens? The tertiary follicle further grows into a matured follicle called Graafian follicle. Here again, theca externa interna will be there, granulosa will be there, the antrum will develop further. Okay, and inner to the antrum we find the secondary oocyte with first polar body. Okay, and this secondary oocyte has already started the meiosis 2 but unable to complete it, so remain arrested in prophase 2 of meiosis 2. Okay, and it is mainly because they lack centriole. Okay, great. Apart from that, the zona pellucida, corona radiata, and cumulus ophorus, this all structure we find, and they remain attached with discus proligerus. Now, once the Graafian follicle is formed, the process of ovulation occurs. Okay, and this usually occurs at day 14 of the menstrual cycle. In this process, discus proligerus ruptures. Okay, the wall of the ovarian follicle ruptures, the wall of the ovary ruptures. As a result, the internal structure having secondary oocyte, first polar body, corona radiata, and some cells of cumulus ophorus are released. Now, this whole released structure is called ovum, right? Great. Now, once this ovum is released from ovary, it will be captured by the fimbri of the fallopian tube and it will enter into the ampulla. Okay, first infundibulum and then ampulla, it will go on. We will see that part later. Okay, now the part of Graafian follicle which is left into the ovary, okay, mainly consisting of follicular cells and the stromal cells, now they will transform into corpus luteum, which is a type of endocrine gland. It functions as an endocrine gland and it secretes a particular female sex hormone in large quantity called progesterone. Okay, if pregnancy does not occur, in that case the corpus luteum will be degenerated in next 14 days and the scar tissue of corpus luteum is called corpus albicans. Great. Now the ovum released, okay, this has corona radiata and the secondary oocyte, zona pellucida, everything. This will meet the sperm. If it meets with sperm, fertilization will occur. Okay, and when fertilization will occur, the cytoplasm of the sperm, okay, containing centriole as well as the male pronuclei and scanty amount of cytoplasm will be released inside the secondary oocyte. Okay, now with the help of the centriole from sperm, the secondary oocyte will be able to complete its meiosis 2. Okay, and again this meiosis is unequal. As a result, two unequal daughter cells are formed. The larger cell is called egg cell and the smaller cell is called secondary oocyte. Okay, and finally, once the egg cell is formed, now the egg cell cytoplasm also has the male nucleus, that is the sperm nucleus. Now the two nucleus will fuse, syngamy will happen. Okay, the process called syngamy and zygote will be formed. Well, so this is how it happens, and this whole development of ovarian follicle occurs in the ovary. Great, yaad rahega. Now, key points to remember here is primary oocyte is diploid, similar to primary spermatocyte. Primary spermatocyte and primary oocyte both are diploid. Secondary spermatocyte and secondary oocyte both are haploid. याद रहेगा, please याद रखिएगा इस बात को. Okay, great. Major difference here is from a single mother cell or from a single oogonia, there is only one female gamete, only one functional female gamete, that is egg cell, is formed. Okay. However, in case of a spermatocyte or spermatogenesis, what we have found is from one primary spermatocyte, okay, there will be formation of 4 spermatozoa or sperm. Okay, so you can correlate with the plant reproduction as well. From one pollen mother cell there will be 4 pollens produced, but in case of female, okay, from one megaspore mother cell only one functional megaspore was developed. Similarly, in case of animals, from one primary spermatocytes 4 sperms, that is 4 male gametes, are produced, but from one oogonia only one functional female gamete is produced, one egg cell is produced, right? Remember. So if somebody asks a question as how many divisions, cell divisions are required for formation of 100 human embryos, right? The answer would be 125. Okay, why? Now you can calculate or correlate from the plant reproduction in flowering plants. Okay, great. So this was the whole process of oogenesis in human female. Okay, great. In next class we will proceed from here. Okay, and we will look at the process of menstrual cycle, which is very crucial. Okay, so we will take an elaborate look on the menstrual cycle, especially the role of hormones, okay, and remaining all the processes of menstrual cycle. Well, that will be very important. A lot of questions are being asked from the menstrual cycle. Okay, so great. I hope you find this video quite informative. Okay, in the next video we will meet again discussing the menstrual cycle. Till then, take good care of yourself, protect yourself. Okay, sanitization is the key here.